Evening viewer, welcome to our Glory Globe TV viewers, a most amazing news. Professor Christoph Neuhaus and his team at the Institute of Molecular Biology in Mainz, Germany have identified a DNA modifications known as 5-formyl cytosine, that is called 5FC, as an epigenetic switch that activates genes during the early stages of embryonic development. This finding proves for the first time that vertebrates have more than one type of epigenetic DNA mark and sheds new light on how genes are regulated in the earliest stages of development. Their findings were published in the journal Cell. Our bodies are composed of trillions of cells all working together to form a functional organism. Yet each of us started off as just a single fertilized egg cell. To become a whole human being, this single cell must multiply rapidly, forming all the correct organs in the right places. These processes of development depends on thousands of genes being activated at exactly the right time and place. Yeah, the activation and deactivation of genes is controlled by so-called epigenetic modification, that is, chemical groups attached to DNA and its associated proteins that act like traffic lights to switch genes on or off. For decades, scientists thought that vertebrates had only one type of epigenetic modifications on DNA called cytosine methylation, which is associated with gene silencing. 10 years ago, 3 more chemical modifications were discovered in vertebrate DNA, but as they were only present in very small amounts, scientists were uncertain if they were functional epigenetic marks. Now, Professor Christoph Neuhaus and his team have shown for the first time that one of these modifications, 5-formyl cytosine, is involved in activating genes in early development. The discovery is significant because it proves that vertebrates have more than one type of epigenetic DNA mark and uncovers a new, also previously unknown mechanism of epigenetic gene regulation. These findings are a real breakthrough in epigenetics because 5FC is only the second proven epigenetic DNA modification besides methyl cytosine, said Neuhaus, founding and scientific director of the IMB, which was opened on the campus of Johannes Gutenberg University, Mainz, Germany in 2011. In their study, the scientists looked at 5FC in frog embryos using microscopy and chromatography. They discovered that 5FC increases dramatically at the very start of development during a key step called zygotic activation, when many genes become switched on. As Eleutheria parasitica, the first author of the study, explained, the observation of 5FC in microscopically visible tiny dots or chromocenters were exciting. Based on them, we suspected that 5FC must do something important in early embryonic development. Amazing. Behind this exclusive report, Glory Globe TV, Delhi, Bharat.